I showed a table like this last time. There is an option in Minitab which will allow you to see which strains are different. In this case, of course, there are only 2 strains and we showed that they were different, but if you had 3 or more, you'd want to see which strains were different. So, of course, we found they were significantly different, and they've got a different letter here, which, as the note underneath says, means that do not share a letter are significantly different. So we've been able to show the strains are different. Another question you might want to ask, you perhaps already thought this in this study, is, well, the injection effect wasn't significant overall, but perhaps for individuals, is that the case for both strains? Perhaps it was having an effect in one strain of mice, but not in the other strain. So we can address this using something called an interaction. You specify that in the software, usually using a star or a dot. So you put in this term known as an interaction, and that's the term that's going to assess the hypothesis that the effect of the injection was the same for both the strains of mice. So we get out an analysis of variance table looking something like this, and the main difference is we've now got a 3rd row added in to address that interaction, and we find a significant p-value. This p-value is less than 0.05. So this has shown us that the injection does have a significantly different effect on the 2 strains of mice. So that's actually a very interesting finding for this study. Although on average over the 2 strains the injection effect wasn't significant, we are finding that we've got enough evidence to say that the effect size was different between the 2 strains of mice, and we'll want to go on and find out which strain there was a bigger difference for and whether that was actually significant. In Minitab we get this output. This is for the interaction. It's going to tell us for each of the 4 groups of the data. So the CD1 strain who had chlorum compared to the control group for that strain both had this letter A. So that says there's not a significant difference between the effect of the injection for that strain of mice. But for the 2nd strain, C3H, if we look at the control row and the chlorum row, we get an A and a B. So that means there's a significant difference depending on the injection type for that strain of mouse. This has been quite an interesting way to study this. You've managed to study two strains at once and establish that this injection is different. The effect of the injection is different between the strains. It only seems to be having an effect in one strain, and that's only possible really to address using an analysis of variance model that fits the interaction. Of course, you could just take the data for each strain separately, but that hasn't actually proved the interaction. It hasn't proved that there's a significant difference in the effect between the strains. You can only do that with analysis of variance. So we might want to know, well, what are the p-values? It said, that table we had just now said there is a significant difference. The letters are different, but I'd like to know what the p-value was for that test. And usually in packages there's some kind of option that will allow you to guess at that. I think the Minitab output that you get from using the appropriate option is particularly unfriendly, but it does have the information in that you want. You have to sort of read this table quite carefully, work out what it's looking at. It's looking at here the C3H strain on the mice that had the active injection, and then comparing it to the 4 other groups. And the one we're interested in, of course, is the control group for that strain. We don't want to compare it to the groups for the other strain. So I've highlighted that, and that's where we've got the significant effect. So we knew it was significant, but now we know what the p-value is, 0.02. So it's not highly significant, but it's definitely significant, which, given this is a small study, it was quite good to find that. And of course what we also want is to get the p-value for the other strain, and we need to go down to this table where that strain on chlorum is paired to the same strain CD1 on control, and we get a non-significant p-value. So there's no evidence at all that the injection is having an effect in that strain of mouse. So it's been quite an informative study, but we did need analysis of variance to draw the full conclusions from it. Now look at another example of using a two-way ANOVA, because in the last example there were 2 effects we wanted to investigate. In this example, it's just that there's some structure in the data, and it's to show we can make the analysis a bit more efficient by taking that into account. So here we've got cages full of mice, and they are being tested on different types of fluid, five different types of fluid, to see which they prefer, and it's done by looking at what proportion they drink of that particular fluid compared to water. And so it's not measured on an individual mouse level, it's measured on a cage level. So these are the results. So you've got sets of mice in four different cages, and the same mice stay in the same cages throughout the experiment. So you might expect there to be some correlation in the results for the 5 results for cage 1 and for cage 2, because the same group of mice that are in each cage. So this is the proportion of the test fluid drunk when they're given a choice of a test fluid and water in each of the cage. And of course, the water is really a control. You'd expect that to be close to 50% because they've just got 2 bottles of water in that cage. The analysis we might think of is we've got the fluid type, and that's what we're really interested in. And I mean, what you could do is you could just do an ANOVA and put fluid type in it, but that would miss the fact that you've got the same mice in these cages throughout the experiment, and their data might be correlated. So there might be a tendency for cage 1 to have higher or lower values than other cages, for example. That's the model. So we would also put cage in the model. We're not really interested in what the cage effects are, but we are interested in sort of taking it out of the model, getting rid of the variation due to cage, but it could be classed as sometimes people will call it a nuisance effect. We fitted 2 things in the model, so our analysis of variance table has 2 rows, and we're interested in the p-values at the end. Well, we're particularly interested in the fluid type, which is highly significant. So there's highly significant differences in the amounts, in the preferences of the mice for different fluid types. But it's interesting to see that the cage effect is also significant, and so that is going to have made the model more efficient compared to if we had just completely ignored the fact that the same mice are in the same cages throughout the experiment. And if we took that out of the model, we'd find this p-value would go up a bit and it would be a less efficient model. So we've been able to adjust for the effect of cages by using a 2-way analysis of variance. And of course, now we've established that these 5 different types of fluid, the mice have different preferences for them. They like the ethanol and particularly the sucrose, and of course the water they don't really have a preference for because it's against the control of water. So I've given here some output from SAS just to show how packages can differ so much in terms of how they display output. So this is SAS's output. That's another statistical package for comparing the groups. And what it does is it gives a p-value, rather than like the letters, the A's and B's that Minitab gave to show if they were significantly different. It gives you a bit more information. It gives the actual p-value for each comparison of each of the 5 fluid types. And it tells you what the numbering is up here. So we've got 2 is saccharin, and it's compared to 1, which is ethanol. And that's the p-value saying that those 2 fluid types didn't differ significantly. But you'll see that there's quite a few sort of small significant p-values. So there's significant differences between quite a few of the fluid types, which we're expecting because we did find an overall fluid type effect. And you can look at the mean values to see which was the most favoured one. So the sucrose was the most favoured, that's number 4, and that's actually significantly higher than all the other types of fluid. So clearly the sucrose was the preferred fluid over all the other options. Another small thing to note here is because we've got 5 different groups and there's lots of pairwise tests we've been able to do, there's actually 10 significance tests that are being carried out here. If the more significance tests you do, then the more chance that one of them is going to be significant just due to random variation. So you need to bear that in mind. If you want to take more account of that, there are ways of adjusting for what's called multiple testing, but I'll not go into that here, but it's just to sort of bear in mind the more tests you do, then the more likely you are to get a significant result just by chance. And it's for borderline ones, which we don't have many of here, but if you say got a p-value of 0.04 and you've done 100 tests, then you need to bear in mind that 4% of those are going to give false positive results.